everybody. Good to be here, and I've really enjoyed the day. I'll wait a second for the room to settle. So yeah, I did say I'd introduce myself. So my name is Lindsay Edwards. I'm the CTO at Relation. I've been working on the interface between computation and biology for longer than I probably should have been, and on drug discovery since Richard hired me at GSK 10 years ago. Yeah, when I say, actually, first of all, Relation is an end-to-end drug discovery company. We're based in London. We use a lot of GPUs. I've lost sight of Bianca, but to the point whereby NVIDIA actually invested in us in our last round, which is very cool. We raised $65 million. I guess one of the unique selling points of Relation, and I promise you, this is not going to be a 15-minute pitch for Relation. That would be very dull. But we are a fully integrated company. So we have our own labs. We do CRISPR and perturb-seq on site. We run a clinical study to garner data, to train our ML models. So we do a lot of stuff in a very connected way. But I wanted to start, well, first of all, just when I say machine learning and drug discovery, I can almost hear people rolling their eyes, because there's so much of it about at the moment, and so little of it is really making any difference. And I'm right there with you. And Lynn will notice that I don't use the phrase artificial intelligence or AI at all. It drives me nuts, because it's so vague as to be completely useless for a serious conversation. And all of this kind of AI accelerating healthcare and AI accelerating drug discovery is so vague. And I had one of those kind of light bulb moments last week. This brilliant sketch, Monty Python sketch from sort of 50 years ago. It's a fake kids TV show, and you've got Graham Chapman, Eric Idle, and John Cleese, and it's called How to Do It, and they cover how to play the flute. Apparently you blow down one end and move your fingers up and down the outside, and they also cover how to cure all known diseases, and I thought you'd be interested. And basically what you do is you become a doctor, you discover a cure, and then you become really famous and you cure all the other diseases. And it's ridiculous, but this is the level that we're having the conversation at the moment. It's like, well, we'll do AI and big data, and it's not a serious conversation. So the reason I'm starting here is because I'm going to commit now to give you as much detail, hopefully that you could pretty much reproduce what I'm going to talk about. I don't think it's an IP issue, because I've done it and it's really hard. So, good luck. But the other thing that I will say is, there's a lot from my world of machine learning who have started machine learning for drug discovery companies who have never discovered a drug in their lives. And so one of the nice things about Relation is quite a few of us have. Our first indication is osteoporosis. I said to Richard this morning, I feel like a bit of a fraud because I'm not really a longevity researcher, but osteoporosis is a serious issue for about half of us once we pass the age of 50. My mum has very serious osteoporosis. I think we all know the statistics of what happens when people fracture their hips. Actually, it's very fortunate for us because our clinical study takes all of this bone waste, and we're the only industrial lab that can do single cell on it at the moment, and we use it to train models. But it's a huge problem. I took up skateboarding a couple of years ago. I was saying to Gina, and so I know I haven't got osteoporosis at the moment, but we'll see. It's obviously running in the family. So having said, I won't do vague hand-wavy stuff. I'm going to start with vague hand-wavy stuff. So Ehrum's law, far left, I'm going to assume everybody is aware of this. So medicines are really expensive. We're around $3 billion a drug at the moment, and it's sort of plateaued, but it's still really expensive. Healthcare costs are skyrocketing. On the other side of the equation, we're much better and cheaper at getting whole genomes, and I am going to specifically talk about whole genomes. I will do what I said I will do. And also compute's getting much cheaper. Like the amount of compute that you can run in a GPU now is orders of magnitude different to where we were even just a few years ago. And again, I'm sure Bianca, like me, was playing with CUDA years and years ago when it was a horrible nightmare, and now it's like relatively easy to do. I'm going to skip this slide. So in terms of where you can deploy machine learning in drug discovery, the first thing to say is uncontroversial is that the big problem is not really molecular design, it's target ID. And it's a huge problem because you don't really find out whether you found a good target or not until you spent tens of millions of dollars and you give it to humans. But there are ways of kind of breaking the process up, and at Relation, we have kind of three areas of focus. So the first one we refer to as risk, and this is around genetics, and I will talk about this principally today. We also do a lot of large-scale cellular perturbations because context is everything. So we run perturb-seq screens in the lab, and we use generative models to kind of understand how cells respond to perturbation. And then prioritization is about kind of deciding what things actually look like reasonable drug targets in terms of their tox and druggability and all this kind of stuff. And the reason I'm talking about genetics particularly, and again, I'm assuming I'm preaching to the choir, is because one of the rare pieces of good data we have on good drug targets is that if you have human genetic evidence that links a trait associated with your disease to the thing you're targeting, then your probability of clinical success goes up a lot. First round of estimates was twice. I think actually the cleaner estimates now, Matt Nelson just printed an update in Nature, it's about 4x. But it's a really, really good signal, and it's fairly reliable. And there is some emerging evidence, it's still very early, that single cell gives you some additional signal, and also that it's somewhat orthogonal to the genetic data. It wouldn't really matter if it was the same signal, but it actually looks like a slightly different signal. So we make a lot of use of both of these at relation. So as I said, we have these kind of three stacks. So on the left is what I'm mainly going to focus on today. So we use large language model architectures. We're not using them to kind of generate synthetic text. We're actually using them to understand the structure of the kind of structure-function relationship of DNA. We also use generative models to kind of predict the way that cells respond to perturbation, particularly in combination, because you get this combinatorial explosion of a problem of things that you're trying to kind of some changes in multiple proteins simultaneously driving a disease phenotype. And last of all, we use kind of fairly standard graph neural network, knowledge graph type stuff to understand kind of which from a batch of potential targets are the ones that we should carry forward. And worth saying, we're deadly serious about taking this all the way through. We have molecular discovery programs already running, have targets 2 or 3 running behind them. We have our second internal indication being spun up next year, and we're sort of, in the process of talking to partners about spinning up 2 or 3 more simultaneously. So we're scaling really, really fast. We're not just sitting around training machine learning models. Although we do that too, by the way. Okay. I'll sometimes use the word Roslyn because we have internal code names for models, and the DNA model's called Roslyn. So if I say Roslyn, you know what I mean. So this is building on an existing kind of thread of machine learning research that goes back, I think, to a paper from Brendan Frey's lab in the early 2010s, where he was using then convolutional neural networks to identify transcription factor binding sites. And what he did that was really neat is actually he framed the problem in a way that we can kind of go after it with machine learning. So basically you take a DNA sequence, you slice it up into chunks. We're at 256,000 base pairs at the moment, which I think is more than anyone else, but you drop it into chunks, and then you predict stuff from your chunks. So you can predict things like chromatin marks, you can predict gene expression, which seems like magic if you're a biologist, but it does actually work. So you can predict all sorts of things. We predict kind of everything in Phantom 5. So you've got these large public data sets, we can train foundation models on them, and that's what we do. The kind of additional bit of work I was saying, which it turns out is really hard, is kind of twofold. So the first thing is, in and of themselves, these models aren't necessarily useful for drug discovery. So I'll talk a little bit about how we've adapted them. The second thing is, as I said, context is everything. If you take, I don't know how many people are familiar with the FTO gene and the story around that. There was a variant associated with obesity. It was mapped to the nearest gene, which is this FTO gene. Manolis Kellis's lab, sort of $2 billion or something later, they showed that in adipocytes, it's not affecting that gene at all. It's affecting this one called IRX3 or 4, quite a long further away. It's like 1 million base pairs away. And a couple of things to take from that. One, not the nearest gene, which is what we do a lot of variant to gene mapping right now. And two, you have to look in an appropriate cell type under appropriate conditions to see the effect that you're looking for. So we take this into account, and we're running a large clinical study in osteoporosis patients where we acquire bone from them, and then we get paired whole genome sequence and paired single cell data from the bone, so we can look at osteoblasts, osteoclasts, and osteocytes. I'm not going to go into kind of bone biology now, but we have the actual cells from the patients, and then we can fine-tune the model using those data sets, which then allows us to study the effect of changes in the sequence and how that affects disease osteoblasts and disease osteocytes. So I think it's a really, really important point. You can pull a model off the shelf and you can do stuff with it, but this extra bit to make it useful is actually where all the work is. The way we infer causality is we actually stripped the last couple of layers off the model. We created a synthetic data set using GTEx, where there's a lot of kind of fine-mapped variant to genes. We've actually moved on a little bit from this now, but this was our kind of base model. And what we do is we train the model on effectively a variant, every gene within range, so within 256,000 base pairs, and the model just scores from 0 to 1 the probability that the variant is affecting this gene in this particular context. And it turns out this works really pretty well, and I'll get to some of the biology in a second. In terms of on the kind of standard task of predicting gene expression and chromatin marks, the best model when we entered the market was DeepMind's Informer. We are a little bit better than Informer. I would, however, say please note this tab before. So kind of this is where we tend to get stuck in the machine learning community, is like, my benchmark's better than your benchmark, and so, I can publish it in Europe's. So yes, you can do that, but it doesn't really get you anywhere. And then on the right-hand side, GeneGenie is a Vibra-Ghebs model. Again, we validated that we had a slight performance increment. And again, what I tend to say to people is, this slide, I hope, says we kind of know what we're doing. I'm not trying to say, like, we're better than DeepMind and we're better than... It's like we're all kind of in the broad same place where, we know what we're doing when it comes to the ML side of things, although we are slightly better. On the osteoblast data as well, we went and got some human data rather than yeast. I'm a recovering systems biologist, so I am aware that yeast is different to human cells. So we worked with the University of Kyoto. We ran these massively parallel reporter assays. By the way, genius for generating data for machine learning if you want to get into our game. Because you can generate these huge data sets with kind of randomly varying DNA sequences, and you can have a reporter assay downstream that kind of tells you how the changes in sequence are affecting expression of some target gene. So we generated a lot of data that way, and the model was pretty good there too. Okay, so I want to get into the kind of nuts and bolts of why this is interesting. And one point I want to make up front is, like, I'm going to do a bit of a numbers game. So a bit of like, this is slightly better than the thing that we all do, because that's quite a useful statement, although it's very, very hard to benchmark these things when ground truth is so difficult to get to. But actually what I've kind of come to is, what's much more interesting is, how can I discover biology that you couldn't discover if you didn't work with us? Like, that's much more interesting, and I've got a couple of examples of that. Mainly where current methods almost definitely go wrong, and these methods are right. There are loads of other advantages we're just discovering. I think you, for example, could increase your statistical power in GWAS studies using these models, because they bake in a lot of prior knowledge about how structure in DNA function, DNA sequence affects function, or just on a basic statistical level, what variants are more likely than other variants to cause sort of biological sequelae. But here's the kind of basic numbers of what we're doing with Rosalind at the moment. So we're doing variant to gene mapping, and when you do variant to gene mapping, you have what we kind of broadly split the problem into two. So if you've got a variant and a gene and nothing for miles around, we tend to class that as like trivial variant to gene mapping, because it's that gene. It's almost as easy as if you've got a variant in a coding region. So that's easy, we'll just kind of bank those. And then you get into this business of non-trivial variant to gene mapping. So this is where you've got large stretches of DNA and you've got one variant and you're not sure which gene you're acting through. And just in case you're thinking this is not an important problem, if you're going to get a genetic signal to support a drug target, or if you're going to use genetics to drive a drug program, if you get this wrong, all of your biology will be wrong. So it's really important that you get this right, and we 100% get this wrong. The FTO gene example is an example of people spending billions of dollars because this process was done wrong. So what you see in the histogram is the distribution of genes per variant in osteoporosis. So osteoporosis has something like 1,000 loci. It's a strongly heritable, very strongly genetic disease. One of the reasons we picked it. This is the 500 or so variants that are non-trivial. And you can see it ranges from 2 variants up to nearly 30. So there's variants where you've got 30 potential genes within range. And you can hopefully see where I'm going here. Like 1, trivial, 2, it's a coin flip. Once you get to 20, it's really, really hard to pick the right gene, and yet that gene might be a really, really important one for understanding the way the disease works. One other thing, having said that I'm not going to do the numbers game, if you kind of look at the number of genes you would have to test, because we validate everything, we CRISPR everything out in the lab, we have human cells, functional assays to see if this stuff works. If you want to do that, I'm going to have to look very ancient. Yes, it's 2,000 genes. I couldn't remember the number. So 2,000 genes for the 420 variants. If you say 3 cell types, like osteoblast, osteoclast, osteocytes, we reckon that would take you 7.5 years, and that's with none of the people in the lab ever having a holiday or a day off sick. So there's like no way really that you can experimentally prosecute the entire space. So this is another thing where computation really helps you do something that you wouldn't be able to do otherwise. So, in terms of raw numbers, if you look at everything over 2, the model is about 50% better than the current best methods that we use. And most of the best methods that we use are related to this idea of nearest gene. Again, it's somewhat controversial how good nearest gene is. So in this case, I'm just assuming that if you go with nearest gene and there is a 1-to-1 mapping between a variant and the gene that it's mostly acting through in this disease context, again, all of these are open to debate, the method is going to be wrong about 50% of the time. It's not quite the case because there are more tumors than there are fibers, if that makes sense. But broadly speaking, Rosalind's about 50% better. Once you go past 2, the numbers really start to kick up. And that's because if you imagine, again, the number of genes means that your method that's strongly indexing on nearest gene means that your hit rate goes down more and more the more possibles they are. It's just basically kind of probability theory. And so in that case, Rosalind continues to maintain its accuracy, and we're up about double, I think, once you get to 3 and above, and that gap continues to increase. I want to give you a concrete example. I'm afraid I've removed the names of the genes to protect the innocent. But in this case, we have a variant. This is a variant strongly related to osteoporosis. It has 10 genes within range of the lead SNP. So again, it's a non-trivial experiment to look at these and then for every single variant. What you can see in the bottom right-hand panel is our human phenotypic assay. This is a calcine assay, so this is human osteoblast laying down bone in a dish. And you can see on the left-hand side there, that's the gene that Rosalind predicted with a high degree of confidence, and it's close to our negative control. So it was a really strong hit. And I promise you, this is not an isolated example. We have plenty more of these. And it's really heartening for us to see the model doing something. And like I said, the key thing for me here is like, this is a gene that wouldn't get picked otherwise. So for us, it's really exciting. You can look at this and go, okay, new biology, maybe there is an opportunity for a therapeutic here. I want to talk quickly about acquiring data to train the model. So we're running a study called Osteomics. Osteomics is 200 and some. The reason I don't know the number is because it's traveling at speed. I think it's sort of 2 patients a week, so I would have to check exactly the numbers, but more than 200 patients. As I said, we're the only industrial lab that can do single cell on bone at the moment. We collaborate closely with partners. I'll talk about that in a second. We're continuing to recruit, and the way that we use this data is we fine-tune the model with paired whole genome sequence and then paired single cell for patients. And as I said before, the aim here is to take a foundation model and contextualize it specifically to disease. So you have a much better chance of understanding how a specific variant is affecting biology in a patient osteoblast. In practical terms, we're about 140% new biology from using this model. And in real terms, again, I've had to remove some of the names to protect the innocent. Obviously, there's quite a lot of IP behind this slide. But we're recovering an awful lot of known osteoblast biology. We're also recovering a lot of novel osteoblast biology. And as I said before, we're pushing this forward into real drug discovery programs. So our first program is up and running. We're at a molecular discovery phase. So it's we're putting our money where our mouths are. So hopefully that's enough detail to kind of believe that there is an option here for using a machine learning model to specifically target something in drug discovery that's truly useful. There are other examples of other things, and so please sort of tap me up on LinkedIn or come and talk to me afterwards. I'd be really happy to talk you through some of the other stuff we're doing. The perturbation stuff is really exciting, also really, really difficult. It's a big area of activity in the field at the moment. In terms of the company, one of the key things that we do is, as I said, we generate a lot of our own data. We call that lab in the loop. This is mainly a reflection both of the fact that we do iterative cycles between the machine learning and what we do in the lab. I think it's really, really important to recognize that biology is much too complex to just kind of have a one and done approach to your data acquisition. UK Biobank, for example, is an amazing resource, but boy, do you ever want to go back and do an experiment and sort of prosecute it. So we do that in-house, and it makes my life as a machine learning person an awful lot easier. We have very strong leadership. Jim, I met for the first time today, but I always recognize him from my slide, so really nice to meet you. So our investors are amazing, and we have a real blend now, particularly since the last round, of both tech investors. So like I said, NVIDIA put money in, but also Deerfield. And so we sort of had that kind of badge of honor from both the hardcore biotech investors and the hardcore tech investors. DCVC, who co-led the last round, also funded Recursion, if you're familiar with Recursion, so they know what they're doing. They're kind of in it for the long term. David, my boss, was the ex-US head of R&D at Pfizer, again, experienced drug discoverer. Ada was a DPU head at GSK. Charlie founded Freenome, if you're familiar with Freenome. So we've got really solid talent and fantastic backing. And the last thing, before I dive away, is we have amazing collaborators. So Michael Bronstein, who's chair here, is one of our kind of key machine learning folk. Caroline Euler at the Broad, Alex Shalek. We have some fantastic domain expertise in OP, like Mark Ween, for example, John Whitaker. And it's worth saying, kind of, these are not faces on a slide. So Mark was in the office the other day. I talk to Caroline and Michael regularly. These are people who are kind of deeply involved in what we do. And it's really important. David likes to remind me from time to time of Joy's law that most of the smart people work somewhere else. So this is kind of one way of allowing ourselves to tap into the broader ecosystem. And like I said, I'm hopefully very approachable. Please sort of tap me up on LinkedIn or come and talk to me afterwards. And I would be delighted to tell you more about what we do. And last but not least, we have a very, very strong focus on patients because it's kind of what we do. Our sort of company tagline is "The patient is waiting." We had an amazing OP patient come and talk to us at our first offsite the other day. And with that, I'm going to stop and see whether you've got any questions. But thank you.